Hello. Good morning, everyone. Espero. No good morning yet. Yeah. Un poco incómodo. So good morning, everyone. Welcome back to the seminars of the museum. Today, it is my pleasure to introduce here the invited speaker, María José Ruiz López. Many of you know her. She did a PhD here at the museum in the Department of Evolutionary Ecology. And then she spent 8 years in the United States, working as a postdoc at the University of Missouri and the University of Oregon. And after that, she came back with the Marie Curie Fellowship in 2018, back to Spain, working at the Estación Biológica de Doñana in Sevilla. I forgot to say that during her postdoc, she mixed her work on conservation genetics and disease ecology using genomic tools. And what she's working on at the moment, we will know now. She in this Berlin talk. Thank you, Marie.
Thank you, Iker, for the invitation. It is a pleasure to be back at the museum, especially with all these familiar faces and friendly faces. So I'm going to talk today a little bit about how combining genetics and genomics with how combining genetics and genomics with ecology can help us understand and explore better host vector pathogen interactions across different scales, from the individual scale to the ecosystem scale. It is not moving. So emerging infectious diseases have been a constant in human history. The Black Death, the Spanish flu, AIDS, and more recently, COVID. And what do many of these diseases have in common? They are all zoonotic, is to say, they come from animals. In fact, 75% of all emerging infectious diseases in humans are zoonotic, and 60% of all human diseases are also zoonotic. On top of that, 17% of those are vector-borne. And there is a common problem. Both emerging infectious diseases and vector-borne diseases pose an increasing threat to global public health. I don't know why this is moving faster. Okay. So the United Nations have identified that the deterioration of ecosystems is one of the main causes of this increase in emerging infectious diseases and vector-borne diseases, and has identified 4 main drivers of this increase. First, climate change, then land use change derived from agriculture and livestock use, habitat fragmentation, and the invasive, I don't know, and lastly, species trafficking and animal movement. But it is not only human populations that are affected by emerging infectious diseases and both vector-borne diseases. They are also an enormous threat to wildlife. Here, we have 4 of the main examples of how emerging infectious diseases and vector-borne diseases can drive populations to extinction. We have the chytridiomycosis, who has caused the decline across the world of amphibian biodiversity, the white nose syndrome, caused by an invasive fungus that killed 6 million of bats in the United States from 2006 to 2010, the devil facial tumor disease, which is caused by a virus and is endangering more an already endangered population, like the Tasmanian devil, and then finally, the avian malaria, which has caused the decline of Hawaiian honey creepers. It is caused by the invasive parasite, Plasmodium, who has entered the islands together with an invasive mosquito, Culex quinckia fasciatus, and that has driven the extinction of honey creepers. Therefore, one of the main challenges that we have right now to understand infectious diseases and understand host pathogen interactions is to do that in a framework of this changing world. And to do that, it's more important than ever to look across different scales and use new technologies. We have the individual scale where the interaction between the parasite and pathogens and the host vector depend on the virulence and immune defense, for example. Then we have the population level scale where we have the abundance and the contact between the different individuals, and that is going to modify the transmission modes and the transmission rates of the pathogens. Then we have the communities where we have different reservoirs, different hosts, vectors, etc. And these community interactions are going to affect parasite transmission. And finally, we have the ecosystem level, where we also are going to have the environmental factors like climate change, pollution, urbanization, habitat loss, and globalization affecting this parasite host environment. This is especially relevant in vector-borne diseases because often they have multiple hosts and multiple vectors that can transmit it. And this is going to affect the ecology and evolution at the individual level and population level of the interaction between pathogens, vectors, and hosts. Genetic tools are very useful and they are like fashionable nowadays, but there is true that they can really help us go a little bit farther into the understanding of host pathogen evolution. We can use population genomics to understand the host genome and population structure of the both parasites, hosts, and vectors. With functional genomics, we can understand what are the genes underlying host defense, vector defense. We can carry on disease surveillance using metagenomics. And we can use phylogenomics to understand parasite evolution, host evolution, vector evolution, and track the epidemiological pattern that they have and the epidemiological contact tracing. We can also it can also help us understand the parasite origin and spread and parasite transmission routes. And landscape genomics is going to help us understand what are the environmental factors that are limiting the movement of the different parasite, host, and vectors. So altogether, they combined with ecological perspective, they can help us look at different scales. So today, I'm going to talk about 3 different study systems that have 3 different questions that goes from genes to ecosystems. First, I'm going to talk about the impact of habitat fragmentation on avian parasite communities in a tropical area. Second, I'm going to stop using this. Second, I'm going to explain how we can look at what is the response of cool excipients to different avian malaria lineages from a genetic perspective, which genes are involved in this relationship. And I'm going to end with one of the zoonotic diseases that is currently one of the major problems in Spain, that is the West Nile virus. And I'm going to explain what is our work with West and how are we identifying which are the factors and the transmission dynamics of West Nile virus in Andalusia and also Europe. So going with the first example system, the goal of this study is to analyze the impact of land use changes on avian parasite communities on the São Tomé Island, and it's a collaboration between Sevilla, Instituto de Salud Carlos III, and Doñana Biological Station. So our study area is the island of São Tomé, that is in the Gulf of Guinea in Africa, and it is a tropical forest that is being very damaged because of palm plantations. So palm plantations are leading to habitat fragmentation in the São Tomé Island. This is causing an incredible amount of deforestation, and this deforestation brings both abiotic changes and biotic changes. These biotic changes include fragmentation of the landscape, the land cover type changes because the primary forest changes to a more degraded forest. If it recovers, then it changes to a secondary forest. And also the area of forest changes. With habitat fragmentation, the area of the different fragments of forest is going to decrease. And that is going to have an impact of disease transmission. All of these changes, both biotic and abiotic, lead to biodiversity loss and biodiversity homogenization of species, and also changes in the ecological interactions and transmission dynamics of parasites. So what we did to understand how does land use change affect habitat parasite communities, so we sampled in the south of the island in an area where we have a mix of habitats. We have a tropical forest, we have a border where we have forest, secondary forest mixed with palm plantations with the borders, and then monoculture of plantations. This can give us a good anthropization score where we can understand how parasite communities change across it. What we did was sample 4 species. 3 of them are endemic, and the blackbird olive thrush. This one is distributed all across Africa. We brought the blood and then did DNA extraction, and in this case, we used a metabarcoding approach to identify multiple families of parasites at the same time. What we did was preparing libraries that were specific for different parasite families, and we also used primers to block the amplification of the birds. And then we used upscale pipeline to identify the different OTUs, and we reached to family level, and we were able to identify up to 5 different families of parasites. So the 3 that were more common were Eimeridae, or Coccidia, Filaridae, where we have nematodes and Dirofilaria, and Plasmodidae, where we have the different avian Plasmodium, the 3 of them, Leucocytothon, Plasmodium, and Aemoproteus. I'm going to focus on the results for Eimeridae and Plasmodidae because Filaridae didn't have, the prevalence of Filaridae didn't have a relationship with habitat fragmentation. It was associated with the species of bird and whether the season was rainy or not. So I'm going to focus on Eimeridae and Plasmodidae that had changes in prevalence and presence depending on the habitat. So first, for Eimeridae, they were more frequent in forest habitats. This result was consistent both looking at presence-absence models and also beta biodiversity and environmental fitting. So what we found that was among individuals where Eimeridae was present, approximately 60% of them were from low anthropization habitats. And Eimeridae was found about 36% of individuals living in these habitats. The species of bird that carried more coccidia were the Proteus santichomae, which is an endemic bird that only lives in the forest fragments. In contrast, Plasmodidae were more frequent in plantations. So they were more abundant in those birds, like the Turdus olivaceus, that lived in the monoculture. And in particular, this relationship is probably related to a higher abundance of Plasmodium, which are transmitted by Culex mosquitoes. So we have 2 different patterns of distribution, and the habitat fragmentation is affecting them differently. So these different effects of land use change on parasite load might depend on the transmission cycles. So Eimeridae, and I have here the example of Isospora, which is one of the more common in Passeriformes, is transmitted through the fecal or root. In contrast, Plasmodidae is vector-borne. It's a mosquito to be transmitted. So the cycle of Plasmodium in birds is a Culex mosquito that is infected with a sporozoite. It transmitted to birds, and then an infected bird transmitted to a mosquito, and etc. So how is landscape changes associated with different results considering these 2 different fecal routes? So we have to consider what happens with fragmentation. So one of the things that happens with fragmentation is that it leads to smaller patches. If you have a species like the Plotheus santichomae that still prefers to live in the forest, you are going to have, at the beginning of that fragmentation, you are going to have more individuals in that forest. That increases the chances of those individuals encountering other individuals. So the fecal-oral route and the aggregation of the individuals would lead to a higher proportion of coccidia in the forest, although it's supposedly the healthy habitat. In contrast, mosquitoes, in particular Culex mosquitoes, can adapt to open habitat and the plantations because there are water resources where they can breed. In this case, for those species that can adapt also to live in plantations and gardens and habitats that are more modified, we are also going to find more Plasmodidae. We are going to find more species. So that is showing us that there is a different effect, that shows us that there are different effects of land use change on parasite loads depending on the evolutionary history and ecology of the parasites and the differences in the transmission cycles. But avian malaria is not only present in tropical islands. It is distributed all across the world, like you can see here in this map. One of the characteristics that avian malaria has is that it has a high phenotypic and genetic diversity. There are 3 different species that are considered that are avian malaria, and on top of that, there are over 3,000 different genetic lineages. The species of mosquitoes that transmit, that have been identified to to carry more Plasmodium lineages are Culex pipiens and Culex perexiguus, especially because they are here in Europe and we are study them more. But one of the interesting things is that because they are here, we can study the variation and the relationship between Culex mosquitoes and avian malaria. So as part of the master thesis of Ana Martin, which studied what biotic and abiotic factors shape Plasmodium communities in Culex pipiens and Culex perexiguus mosquitoes, Ana identified a high diversity of Plasmodium species in both of them. She identified 22 different species in Culex pipiens and 30 in Culex perexiguus, lineages. One of the things that is interesting is that when we did beta diversity on these species, what we found is that the community composition for Culex pipiens and Culex perexiguus is different. This is led by 2 causes. One of them is what they are feeding on. They might feed on different birds, and this might lead to differences in the lineages that they are carrying. But it also depends on the genetic makeup of the mosquito and the relationship that the mosquito has with the different pathogens and how the mosquito responds to these different pathogens. So the mosquitoes also have an immune response, and parasites also affect mosquitoes and can kill them. What is the basics of the mosquito's immune response? So the mosquito immune response, one, they are feeding, the blood forms a peritrophic matrix, and this peritrophic matrix protects the interior of the gut from debris and other parasites. So how the mosquito reacts to the formation of the peritrophic matrix, it's a very important step in the immune response. There are digestive enzymes that are going to be destroying this peritrophic matrix and also are going to help us in the immune response against the bacteria and parasites that are within the peritrophic matrix. And then the last barrier is the innate immune response that is going to act once the parasites and pathogens that were here are released into the gut of the mosquito and try to enter the mosquito cells. As part of the PhD thesis of Marta Garrigós, which I am supervising, we studied what is the transcriptomic response of Culex pipiens to the infection of two different Plasmodium species, and Plasmodium relictum or Plasmodium catamerium. The goal was to understand if the immune response of the mosquito was changing depending on what parasite they were infected from. Supposedly, Plasmodium relictum and Plasmodium catamerium are very similar, but they might release a different immune response. So what we did was capturing birds in the wild, then we took them to the Doñana biological station, and we allowed Culex pipiens mosquitoes that we had raised in the lab as well to feed overnight on birds that were infected with Plasmodium relictum, birds that were infected with Plasmodium catamerium, and birds that were not infected. And then we did take mosquitoes at 24 hours, 10 days, and 21 days post-exposition, and we did RNA-seq to look at the transcriptome and understand what was the response of the mosquitoes to the different scenarios. And so one of the first things that we saw was that there was clearly a differential expression at 24 hours post-infection compared to the rest of days. This is because at this time is when the parasites that are in the peritrophic matrix are trying to cross the gut barrier, so are directly interacting with the gut cells. We found as well that the mortality of mosquitoes was higher in Plasmodium relictum compared to catamerium and the control. So more Plasmodium relictum infected mosquitoes were dying. The next thing that we saw was that there was a differential expression of more than 1,000 genes between Plasmodium relictum and infected mosquitoes and Plasmodium catamerium infected mosquitoes at 24 hours. So the immune response of the mosquito to both parasites is different. But how is it different? So the differentially expressed genes were associated with the innate immune response, that was the third layer that I was talking about before, and also with the chemiotrypsin, trypsin, and serine metabolism, which are the proteins that are involved in destroying the peritrophic matrix. In regard to the innate immune response, what we saw was that Plasmodium relictum activated the toll-like receptor pathway. This is consistent with the only other two studies that there are in other Culex species, Culex quincea fasciatus. However, Plasmodium catamerium did release an innate immune response, but it wasn't the toll-like receptor pathway. It was a different pathway that is not very clear how it actually works. For the toll-like receptor pathway, it is conserved across different taxa, and it is very known. Then the next difference was in the metabolism of the proteins, digestive proteins. So in Plasmodium relictum, they were under-regulated, while in Plasmodium catamerium, they were over-regulated, all of them. It has been shown in mosquitoes, Aedes mosquitoes infected with Plasmodium gallinaceum, that it also activates these proteins, and it leads to a higher infection rate of Plasmodium gallinaceum to Aedes mosquitoes. So this might mean that Plasmodium catamerium is activating an immune response that allows it to cross better the gut border. However, Plasmodium relictum is activating a stronger immune response. Thus, the interaction between Culex pipiens and Plasmodium is species-specific and is reflected in a different response of the mosquito to each species, which is telling us that we have to go a little bit further. So there are the relationship between, in avian malaria, between host, parasites, and the different Plasmodium parasites that they are having. It is very complex, and this high diversity in lineage, this high genetic diversity leads to a phenotypic diversity that goes beyond just the shape of the Plasmodium that we have. It also is going to trigger a different immune response, and probably they are behaving differently within the mosquito. So all that diversity that we are seeing is translated into phenotypic differences that are related with virulence. Now I'm going to switch gears a little bit, and I'm going to talk about the West Nile virus, el virus de Nilo Occidental. I don't know why I didn't translate it here, but you all know about it. So the West Nile virus is a zoonotic disease. In its enzootic cycle, it is transmitted between Culex mosquitoes mainly and birds. But occasionally, Culex mosquitoes that are infected bite humans and horses and other vertebrates, and humans and horses develop a disease. Humans and horses are dead-end hosts because they do not develop enough viremia in the blood to infect mosquitoes. And why is West Nile virus interesting? Well, it is the most important emerging infectious diseases right now in Europe, and probably in Spain if we don't count COVID. So what has happened recently? So even though West Nile virus has been known to circulate in Europe since the 50s, in Spain since the beginning of the 2000s, we only started to have cases and outbreaks in the 2010. This year, something changed, and what happened was that both across Europe and in Spain, we started to have more and more cases. In Europe, we had outbreaks in 2018 and 2022 caused by both West Nile virus lineage 1 and lineage 2, and the virus has right now spread to Moscow and Berlin. So it is both increasing in number, but it's also spreading towards the north. What has happened in Spain? So in Spain, until 2020, the cases were occasional in blue. We had more horses cases in yellow that are used as surveillance, but the cases in humans were scarce. However, in 2020, we had our first big outbreak. So you have an idea of how important this is. This was the first outbreak of a vector-borne disease in Spain since the eradication of malaria in 1964. So this means that the scenario that we have in Spain has changed radically regarding vector-borne diseases affecting humans. Unfortunately, this year, in 2024, we had another outbreak that was even bigger than the first outbreak in 2020. So I'm going to tell you a little bit about what is the transmission in Spain, in Andalusia, and what are the tools that we are using to understand it better. So to understand the spatial and temporal factors influencing West Nile virus transmission, we had to look at this interaction between vectors, reservoirs, and accidental hosts. In the case of where these come together, we have the high-risk area for the spillover to occur. We have the impacts of the environment, the temperature, precipitation, humidity, socioeconomic factors affect the accidental host, depending on where people live and the capacity that people have to control vectors. The spatial distribution, diversity, and abundance, phenology and migration, and genetic makeup of birds has a strong impact, and so does the spatial distribution, abundance, phenology, and genetic makeup of vectors. So a little bit of background. What we know is that right now is that higher winter temperatures lead to an increase in West Nile virus seroprevalence, both in birds and horses. So thanks to long-term data collected from 2003 to 2020, we have been able to analyze what is the seroprevalence of West Nile virus in birds and in horses. And we have seen that the winter temperature is the leading factor, and the minimum temperature over the year, that leads to an increase in West Nile virus cases in the next summer. In case of horses, precipitation also has, annual rainfall also has an impact and how distributed it is, and that is because of the way horses behave around waters of body. So sometimes, even though when it rains, it doesn't rain much, horses go to the same waters of body waters where birds and mosquitoes go, and that increases the chances of them being infected with West Nile virus. What do we know about the 2020 outbreak and how the spillover in humans occurs? So we know which species of mosquitoes are leading this spillover and are amplifying the virus in West Andalusia. And this is very important. This is the case for West Andalusia. West Nile virus can be transmitted by over 60 different species of mosquitoes, and it has more than 300 different birds that can act as reservoirs. That means that even though to prevent West Nile virus spread, we need to think global, we need to act local, because what happens in each region, the pen is different to what might happen in another region, okay? So in southwest Spain, we have the mosquito Culex perexigus that likes to live in rice fields. It is not distributed towards the north. So out of all the positive pools for West Nile virus that we have, 90% of those are Culex perexigus. This means that this species that often lives in rice fields is increasing and amplifying the West Nile virus. A smaller proportion of positive pools, 8.4%, are Culex pipiens, which is the common mosquito, the house mosquito, the one that comes and bites us. So what happens then? What we think, our hypothesis, is that the Culex perexigus mosquitoes that live in natural areas and rice fields amplify the zootic cycle in birds because they preferably feed on birds. But when there are enough cases and they get closer to areas and suburban areas, what happens is that Culex pipiens that lives all over the place, both in natural areas and urban areas, Culex pipiens might feed on both birds, but if there are not birds, they also feed on humans. So Culex pipiens is possibly the vector that is causing the spillover from birds to humans. So it is very important to control Culex pipiens populations in urban areas, and it is very important to control Culex perexigus populations in rice fields. One of the things that happened in 2020, you might remember, is that we had a COVID outbreak, right? So what happened with that COVID outbreak was that we were at home, and then we had a West Nile virus outbreak. So there was this time where we could only walk around our municipal area, right? So one of the hypotheses to explain why the outbreak occurred in the areas that live close to the rice fields was that people, in order to walk, because they were tired of being at home, would go to the rice fields to walk. And that was why they were infected with West Nile virus. So what we did was, so here, like, to make this more clear, it was like the natural areas were to blame. So this was all a problem with the natural areas. So what we did in 2020 was also look at the seroprevalence, the antibodies of West Nile virus in the urban birds. And we found that they were indeed infected. So they had been infected. So 14.7% of house sparrows, turtles, blackbirds, 91.3% of blackbirds were actually seropositive for West Nile virus. That means that almost every single blackbird that we sample had West Nile virus, which means that it is a very good amplifier. And it also means that humans got infected within the urban area. They didn't have to go to a natural area to be infected. So this is very interesting because actually West Nile virus has been studied for a long time in the United States because it's an invasive disease that is spread in very few years from the east to the west. And in the US, the American robin, which is also from the Turdus genera, it is also driving West Nile virus infection in humans. They are migrant, and when they amplify in an area the virus, and when they leave, the infected mosquitoes, they don't have anything to feed on. So then they feed on the humans. So a similar relation, even though we have a resident, is probably happening with our turdus. So there might be a genetic basis to why turdus are so good amplifiers, amplify so well West Nile virus. And here is where we can use genomics and go a little bit further. For example, we can test, looking at the birds, we can try to understand what are the genetic bases of differences in susceptibility of West Nile virus. Why there are some birds that are very good amplifiers and why others aren't. For example, in a previous study with infected birds, they discovered that chickens are very bad amplifiers and they barely get any viremia at 3 days post-exposition. Meanwhile, Passeriformes, they get at 3 days post-exposition, they are the ones that have the highest level of viremia, and they are very susceptible to West Nile virus. So Passeriformes are the best amplifiers for West Nile virus. So we would have liked to do an experiment with the blackbird, but that's very difficult, so we had to focus on other species. So there are only 2 other studies that look at differences in response of birds to West Nile virus, looking at transcriptomics. So we have zebra finch and house sparrow. They respond differently to West Nile virus. In regards of mortality, house sparrow have high mortality, zebra finch low, and the European turtle dove has no mortality at all. They have high viremia, house sparrow, and high competence, while the zebra finch has medium and moderate competence, and the turtle dove has low competence. So what we aimed to do was trying to understand what was the genetic basis of those differences, why some species are more susceptible than others. And we took the opportunity that there were already 2 studies on the Passeriformes and did an experimental approach with the turtle doves. So what we did was in the P3 of the INEA, we did an inoculation experiment where we inoculated an experimental group of turtle doves, female turtle doves, with West Nile virus, and then we had a control group. At 3 days, 8 days, and 15 days, we took blood, and at 3 days, we also took the spleen from some of them to look at their response, and we analyzed the transcriptomes. So the first of all, what we saw was that the viral load was low. The viral load was not very high, but enough probably to lead to an immune response. And in fact, both in the spleen and the blood, we find differential expression 3 days post-infection. So there are different genes expressed in the birds infected with West Nile virus compared to the birds infected that are not infected. So what are the genes involved in this? So we go again to the innate immunity. In this case, again, toll-like receptor. That was also the one that was responding to Plasmodium in the mosquito. So the activation is higher, overexpressed in West Nile virus infected birds for toll-like receptor 4 and the TRAF6 and all this pathway. In addition, also the autophagic pathway is overexpressed. What is this pathway? So this pathway, it's involved in destroying the viral particles. So we have a response, an identification, and an action to kill those viral particles. The role of innate immunity has been shown in the 2 other studies of birds, and it has been shown in mammals. And the role of autophagy, it's to say the activation of this pathway that kills the virus, has been only shown in mammals. And why it is important? Because what they think that happens in mammals is that those individuals that have higher expression of the autophagy pathway, they don't get sick because the viral particles are destroyed. When the autophagy pathway is underexpressed, what happens is that those viral particles can spread through the body and cross the brain barrier, blood-brain barrier. That is what leads to disease and eventually death. This is done in human models. So what did our study and gene expression analysis have in common with the other two studies? Well, in all the three species, there is an increase in the activation of the innate immune response. However, only in the European turtle dove, there is an activation of the autophagy. And this might explain why eventually the viremia of the European turtle dove is lower than the viremia in the other species, because there is a pathway that is actively destroying these viruses. But genetic approaches can help us understand other things. For example, as I said before, they can help us understand the diversity and strain circulation, that it is not, and that can help us understand how the virus is moving through the space. So to use this, what we did was sequencing genomes from whole genomes from the virus, in this case from the lineage 1. In Spain right now we have 2 lineages circulating. The one that has been circulating is lineage 1, and lineage 2 has entered in 2017 from Europe. So it is moving from Europe. So I'm going to focus on lineage 1. So what I did was use phylogeographic and phylodynamic approaches to understand the dispersion and the factors driving this dispersion of lineage 1. So using whole viral genomes, what we found is that there were at least 5 different introductions. Some of these in viral viruses that have entered, lineages that have entered, are now extinct, and 2 of them are currently circulating. The most recent common ancestor is in the year 2000. That is when we started to detect West Nile virus in South Spain. So right now we have lineage 1, cluster 2, WetMed1-3 circulating, and lineage 1, cluster 2, WetMed1-2 circulating. And what's interesting about this is that they have different evolutionary and dispersion histories. So WetMed1-3 is the one that caused the 2020 outbreak. The samples and genomes that we have from the mosquitoes are identical to those that were isolated from humans. But what we can see here is that once it enters Spain around 2013, what happens is that it has been circulating between the south of Spain, Andalusia, and Extremadura. And it has been going from one region to the other region from more than 7 years. And it is endemic. It is not a viral strain that came from outside on 2019 or 2020. So it was already circulating. In contrast, the other variant that is circulating right now, which is WetMed1-2, is a new endemic variant. Why it is newly endemic? So this has a little bit of a different history. So this variant was first detected in 2012. This variant was first detected in 2012, here in purple, and then in 2020. And then it wasn't detected until 2022 and 2023 in a consistent way. All of the samples that we were sampling had this variant. Here in red, what we see is that the variants that are more closely related to the variants we have been detecting in Spain, and those come from Senegal. So what was happening here is that there was a movement of West Nile virus between the south of the Iberian Peninsula and Africa. It doesn't have to be Senegal in particular. It just happened that Senegal has an Institut Pasteur, and then we have a lot of samples from Senegal, right? But it has been moving through migratory birds, in this case, from Africa to Spain. And then occasionally, it has gone from Spain to France and Italy. In Italy, it caused an outbreak in 2021. Okay? So what we have here is that we have two different histories. When we do the phylogenetic approaches to understand how fast it is moving throughout the space, what we find is that the mean dispersion distance of West Nile virus is 144 kilometers per year. And that there is a different role of migratory and resident birds. Migratory birds move it along long spaces, and resident birds move it in small spaces. Also, the European dispersion is associated significantly with the presence of water bodies, which is to be expected because migratory birds might go from one water body to one water body. And this matches what we are finding in the field. So if you look at this map, there are approximately 144 kilometers between each of these different regions. This is what we have in 2022, where we have 33% of the wet met 1.3 and 60% were wet met 1.2. And if we move forward, we are seeing the increase of the second variant, right, that is moving. And then in 2024, all of the variants that we found were wet met 1.2, and they even reached the area of Cordoba. So what does it mean? Well, first, that in Andalusia, we have lost one of the variants. But we know that it's circulating in Extremadura, so it might come back. And these results show the complex dynamics of West Nile virus circulation, with introduction of new variants, with migratory birds, extinctions, spread, and co-circulation that depend sometimes of microenvironmental changes, which species are bringing it, and the distribution of vectors. And to finalize, I would like to talk a little bit about how we can use genetics to understand a little bit more our vectors. In this case, I'm going to focus on genetic structure, because we actually didn't know what was the role in movement of the mosquitoes. Is there a strong genetic structure? Are Culex pipiens spread all through the way and there is no population structure? There is no adaptation to different environments, and that might have an effect on the control. So as part of Sonia Cebrian's thesis, we did the population genetic structure of Culex pipiens and Culex perexigus, but I'm going to focus on Culex pipiens here, which has no population structure whatsoever. So this means that technically, all the Culex pipiens that we have are similar, and there is a strong gene flow between the different regions. However, when we look at the chromosome level, we found population structure in chromosome 1. So there is actually a difference. We have now confirmed that this is an inversion in chromosome 1 here, where these peaks show the limits of the... No sé por qué se mueve tanto. Where these peaks show the limits and the areas where it is crossing, and this region here is the area where it is, there is recombination. There is a small inversion only also in chromosome 3. And this is really important and exciting results, and I wanted to kind of close the talk with this, because this opens up a lot of new research. So the evolution and ecological importance of chromosomal inversions in Culicidae is only known in Anopheles, where it has been shown that adaptation to chromosomal inversions is important, and it affects insecticide resistance, behavior, resting and biting behavior, wing size, so phenotypic external physical characteristics, and reproduction patterns, like associative mating. So this opens up a new area to explore, because we now need to know which genes are in that inversion, and if that is going to have an influence on how Culex pipiens is transmitting West Nile virus or responding to insecticides. So as a take-home message, I would like to highlight that an integrative approach that includes genetics and genomics and ecology can help us understand the connection between environmental, human in this case, and animal health. And it also allows us to process, to study processes that occur at different scales, from individuals to ecosystems. And this, of course, is the work of a lot of people. from the Jordi Figuerola's group, Ana Vázquez group, and Miguel Ángel Jiménez at INEA, and all the students. So thank you.
So we have now time for a couple of questions.
Thank you so much. It was a really nice talk. I have two questions. The first one is, sorry, I don't know how an inversion can improve the phenotype. Like, how can you get more resistant to insecticide and this kind of stuff? Do you know any reason? Because an inversion is a change of expression, or?
Yeah. So this is something that we know of this since February, and we have confirmed that it is actually an inversion with another group of researchers like 2 or 3 weeks ago. So we are starting with this. But so what happens is that on top of the inversion, there are also SNPs that are carrying resistance to the insecticide, for example. So what it happens is that it's changing, apparently, and this is from what I've read, it's changing not only SNPs, but also a little bit of the conformation. So it is in certain areas where it is leading to insecticide resistance, by also leading to changes in behavior. So it is associated because you've seen like the pattern is not like an inversion that stays completely different. There is recombination inside them. So there are different things going on top of the inversions.
Okay.
So in the case of Anopheles, the role of inversions has been studied before genomics. So what they did just with like, oh, these are resistant or not with cytogenetics.
Yeah.
So there are a lot of things going on top of like the inversion.
And my second question is, you have shown that.
Another thing. On top of the inversion, that changes sometimes the number of duplications, copy and vary. So there are like SNPs and structural changes that affect insecticide resistance through ways that we still need to study, because copy number variation and structural changes are something that we can reach now with genomics, but we couldn't actually reach before.
Thank you. And the second question is, you have shown that it's going up to Extremadura. Do you have data from Portugal? Because I will guess that there are also detection in Portugal.
Yeah, so in Portugal there is West Nile virus as well, but there are fewer studies than in Spain. So actually there are barely any genomes from Portugal. So this is actually a huge problem. Because, as I said, when I was saying Senegal, it's not necessarily Senegal, it's that we have data from Senegal. But the same happens all through Europe. So Italy has, because it has a lot of problems and they've been doing a lot of research, there are hundreds and hundreds and hundreds of genomes from Italy. Now we have generated over 100 genomes from Spain. So now we have kind of filled the gap from Spain. But other countries need to start doing the same to better understand what is going on. So until recently, for example, for lineage 2, it was very difficult to carry out studies where, like the phylogenetic studies, because we didn't have enough sample size to compare the phylogeographic and phylogeny with the layers of temperature and the layers of climate. And it is because all the holes we have through Europe.
Thank you.
You're welcome.
Thanks. There is a question from YouTube, so I will read it. Elena Riero Higueras asks, she says it's a really good talk, and it's confusing because I see you moving in YouTube.
Okay, I will stop.
She asked if there is correlation between transcriptome and in the samples of liver and blood, if you could see this in 3 species or only in the pigeon, Tortola.
Okay, so The studies from the house sparrow come from spleen. So because there are like 2 studies in house sparrow. One is doing a similar approach to what we are doing, and they use spleen data. And those ones, there is a correlation in the immune response genes that we are seeing. But then there is this very nice study in house sparrow where they are actually at an experimental layer where they are doing they are exposing the birds to light. And what happens is that there is a dysregulation in the immune response when birds are exposed to artificial lights, and that changes the expression pathways. So for one of the studies that in house sparrow, yes, there is correlation.
Very cool, thanks. Who has another question? Anna?
Hi, thank you, Mari. It was a very interesting talk. I was wondering if you could capture also the transcriptional response of the Plasmodium when you do transcriptomics, because that would help you understand what happens between the feedback between the two.
Yes, you can actually do that, even though it is more difficult. And often our problem here is like we knew that the mosquitoes that we analyzed were infected because we did PCR to make sure that they carried Plasmodium once. Because when they feed on animals, not every single mosquito gets infected. Some do, some don't. What happens is that we used a natural system, right? And in the natural system, often the parasite load in the bird is lower than in an experimental approach. So there are studies that carry different groups in Sweden where they are doing an experimental approach from the beginning. So they inoculate the bird with the parasite. When the parasite reaches a certain load, then they put the mosquitoes to feed on them. And with those, they have enough parasites to get the immune response of both the parasite and the mosquito. And there are like, we published this at the end of 2023. There are now a couple of preprints that are looking at the expression of the parasite in the mosquito. And with Anopheles, because that is a more controlled system, there are very nice studies that do single cell and spatial transcriptome, where are telling you what happens in each region, both for the parasite and the mosquito. But unfortunately, we are a little behind with the natural systems. But it is moving forward.
Thank you.
Another question?
Yeah, you said about how the prevalence of parasites changes with habitat fragmentation, for example. So you mentioned about species in sort of forested areas. Is that because as deforestation, for example, happens there in closer proximity, and then there's a higher prevalence?
So this is an hypothesis, right? But our hypothesis is that habitat fragmentation has a lot of different impacts, and a lot of things happen. But one of the things that happen is that those forest patches tend to be smaller. Some are going to be big, but some of them, depending on how a habitat fragmentation happens, are going to be smaller. What happens is that we have this in mind where there is a smaller patch and a species go extinct, but that might not happen directly. So a first step, and we've seen this also in primates, what happens is that most of the birds that are still around, or the primates, go to the same piece of forest, and they live in there until there are no resources, and eventually they go extinct, move to a different fragment, etc. When that happens, you have an increase in population density, and that increase in population density might lead to higher probability of different birds encountering each other or sharing areas of nesting, etc., etc. And that might increase the chances of transmission of those parasites that are transmitted by contact or the fecal-oral route, etc. While in the case of Plasmodium, it necessarily needs the vector. So what happens with the vector in the different scenarios that forest fragmentation causes, what is going to eventually happen with the parasite load in the bird. So that's why there is this interaction between habitat fragmentation and land use change, parasite burden that might not be as straightforward as we think and might be a little different.
Okay, thanks.
Yeah.
Thank you. One last question. No more questions. So now I invite you to come to the kiosk if you want to talk more informally with Mari. And thank you all for coming. Thank you, Mari.
Thank you.